Welcome everyone to our SCIEX Biopharma Vodcast. I'm Roxy.
And I'm Kerstin, and in this series we're going to be uncovering how scientists are rewriting the rules of analytical science.
In today's episode, we're talking about protein characterization and exploring new workflows. Our guest today is Tyler Heibeck from Sutro Biopharma. So great to have you with us, Tyler.
Thank you. Glad to be here.
So before we get started, where in the world are you today, Tyler?
I'm at our main R&D building in South San Francisco, California in the United States.
And what would you be doing right now if you weren't on our podcast?
I would be reviewing a report protocol, is what I was doing before I came down to chat with you.
So what's better, being on the podcast or writing that?
Better, we can put a point of debate, but on the fun scale, this is more fun.
Okay.
On the better, probably, this drug might save a life, so.
We won't hold it against you.
To get to know you a little bit better, we would ask you some very quick questions, and we would like you to answer with the very first thought that comes to your mind.
Mm-hmm.
If you were shrunk down to the size of a molecule, which molecule would you be?
Well, since we're talking about Sutro, the first thing that popped into my mind was STRO2, because that's kind of our hottest molecule right now that's in trial. We're putting a lot of work into it.
What would you be if you weren't a scientist?
I would probably be a musician or a photographer. Would be my guess.
Mass spec or CE?
If I only had one, I'd choose mass spec.
What do you enjoy most about what you do?
Probably I most enjoy the old school analytical characterization. The ones I enjoy most as a scientist are kind of what I'll call the warts. This is the milkweed and the thistle, because those are the most interesting. Mm-hmm. As a person who has to deal with some regulatory stuff and also has a lot to do, I am supporting a lot of different people. Mm-hmm. And I'm really enabling other people's work. So that's rewarding.
Now, Tyler, what are some of the challenges that you face?
Some of the challenges, that varies with the analysis. Some of the most recent challenges is identifying new charge variants. You do an ICIEF usually, or a strong cation exchange chromatography, and you see a new peak. You've done a new batch, you've changed your process a little bit, and something pops up. And you need to figure out what that is. So the challenge is how to quickly figure out if that's actually a problem. Is it product related? Is it, you really can't take much action until you figure out what it is. And that's the job of AD, is to figure out what it is.
Tyler, can you comment a bit on the tools you have available to make these types of analysis?
I'm a mass spectrometrist, so I have a hammer, and everything looks like a nail, but that said, it's a powerful instrument. So we do have several mass spectrometry instruments from various vendors, including a SCIEX 7600. But in AD, you have to target other areas. And you choose that for, maybe they're faster, maybe it gives you some different information. And also we have to consider how transferable is the method. Like an intact protein method on, 𝗥𝗲𝗏 is a very wonderful and powerful method. Good luck bringing it under validation and quality control under a QC environment. So probably an HPLC assay is better. So we have a lot of HPLCs. We make use of SEC, and all of these methods really basically tell you, does it look the same? And if it doesn't, what might be changing? And then if that's important, then you dig in.
Considering all these different analytical technologies you have access to, have you ever found any surprise, something you really did not expect at all after all these years of experience?
Probably the one that gives me the most surprise is, the most unwelcome surprises, I'll say, is sequence variant analysis. To be specific, we have a technology where we can incorporate a non-native amino acid. And as people who work with cell-free and non-native amino acid insertion know, sometimes other things can creep in. Usually we do find a way to solve it, because if you don't, then your product might not be able to go forward.
You might have noticed our series is called Rewriting the Rules of Science. Can you elaborate a bit how you think that would apply to Sutro or to your work?
When you're regulated, it's kind of you can rewrite the rules, but you kind of have to talk to the FDA first. So there's a collaboration there. But in the area of drug development, we're certainly rewriting rules all over the place. We were one of the earliest companies to really push for site-specific conjugation of ADCs, and had some very compelling data that it was actually increased the efficacy compared to the non-site specific conjugation. That's a rule rewrite, if any, that I would say Sutro was one of the very early voices in making that change.
Very cool. Thank you.
All right. Well, thank you very much, Tyler. It's been a pleasure to speak with you, and thank you so much for sharing your insights with us. Thank you.
It's a pleasure.
And also many thanks to our viewers out there. Make sure to tune in to our next episode. And until then, feel free to check out SCIEX.com and keep rewriting the rules of science. Thank you.
Goodbye.
Goodbye.